So in this video, we will be understanding what is the pathophysiology of celiac disease. Now, celiac disease, as we all know, is also commonly known as gluten intolerance disease. But specifically, there's a amino acid component of gluten, which is known as gliadin, and gliadin is the main culprit in celiac disease. So let us look at what happens. Usually, these food containing gluten are wheat, barley, rye, etc. So when you eat gluten, the gliadin component of the gluten reaches up to the small intestine. And when you look at the involvement, it's usually mostly in the duodenum, but of course, jejunum and ileum is also involved, but it's mostly in the duodenum, because of which, when we biopsy patients with suspected celiac disease, it is usually done from the duodenum through endoscopy. So this gliadin, when it reaches the, let's say, the duodenal lumen, the gliadin combines with secretory IgA secreted by the enterocytes, the luminal enterocytes in the duodenum or in the small intestine. Normally, this gliadin secretory IgA complex in a normally gluten tolerant person should be destroyed and excreted in the stool. However, what happens in celiac disease is this complex gets absorbed into the lamina propria. And how does this happen? Is because of this protein known as transferrin protein. Now, as we all know, transferrin is responsible for absorption of iron, but in celiac disease, transferrin also helps to absorb this complex. And another problem with celiac disease patients is, because of some unknown reasons, there's more expression of transferrin proteins in enterocytes. So more number of gliadin secretory IgA complex will be absorbed into the lamina propria. Mind you, we're still talking about the bowel wall. So in the lamina propria, this gliadin is acted upon by an enzyme which is known as tissue transglutaminase, which removes an amide component from the gliadin. So this deaminated gliadin is now just present there, which is taken up by macrophage, and macrophage expresses it in its major histocompatibility 2 complex, MHC2 molecule. So there are specific MHC2 molecules in which macrophages express these deaminated gliadin, and these are known as HLA-DQ2 and HLA-DQ8 molecules. Now, this is the mechanism of our immune system, where a macrophage will take up the protein and expresses it for other immune cells to come and have a look at it. So the first cell that comes into it is the T helper CD4 plus cells, and the helper T cells, they come and they have a look at the deaminated gliadin, and as a response to that, they produce inflammatory cytokines like interferon gamma and tumor necrosis factor. These itself cause inflammation and destruction of the enterocyte. On top of producing inflammatory cytokines, they also activate B cells. And B cells, in response to this inflammatory activation, produces 3 distinct antibodies. The anti-tissue transglutaminase antibody, which is against this enzyme, anti-gliadin antibody, which is against gliadin itself, and anti-endomysial antibody. Now, endomysium is the component of muscle. It is the component of muscle which also forms up the lamina propria. However, we're not really sure why this is formed, and we're not really sure what this does, because there's no significant component of muscular damage in celiac disease. However, tissue transglutaminase and anti-endomysial antibodies are very specific and sensitive for diagnosing celiac disease, and anti-gliadin antibodies can also be used, but with lower sensitivities. All of these effectively cause destruction of the enterocyte. So the enterocyte is now destroyed, leaving more space for more gliadin to be absorbed into the lamina propria and aggravating this cycle of inflammation and destruction. Now, because of this, what we see during biopsy, as I've already said, duodenum is from which we biopsy for suspected celiac disease patients. So a normal duodenum in histopathology should look something like this, with its long, tall villi and short crypts. Sorry. So because of destruction of enterocytes and subsequent destruction of the villi are flattened, is not long and tall. There are longer crypts, and another significant finding that we can see in this slide is the immune infiltration into the villi. So that is the pathophysiology and pathological finding of celiac disease. Hope you enjoyed this video. If you didn't understand anything, please write down in the comments below and I'll be more than happy to help you with it. Thank you for watching. Please subscribe, like and share this video if you found it useful, and have a great day.